Hi, my name is Kaleb Tsegay, and for my honors thesis, I explored the extracellular release of mitochondria in the context of mitophagy in an LPS inflammatory model. Mitochondria are generally understood as having roles in ATP generation and regulating cell proliferation, death, and metabolism. However, mitochondria also have emerged as having a large role in pro-inflammatory signaling. And similarly, pro-inflammatory signaling agents seem to affect mitochondrial quality and function. Mitochondrial dysfunction and the dysregulation of mitochondrial dynamics are frequently implicated in the propagation of inflammation in degenerative, particularly neurodegenerative and acute diseases. There is an abundance of evidence pointing towards the role of mitochondrial reactive oxygen species, or ROS, in inflammatory signaling. Free radicals in ROS are byproducts of mitochondrial oxidative metabolism, and inflammation induced by ROS burden acts as a feedback loop. Mitochondrial DNA is particularly sensitive to oxidative damage, which may lead to the propagation of mitochondrial dysfunction in the presence of ROS, leading to cell death. Normally, dysfunctional mitochondria are degraded via the mitophagy pathway, which shuttles depolarized mitochondria via vesicle transport to the lysosome for degradation. This is shown in Figure 1. As this quality control mechanism helps ensure a healthy population of mitochondria to maintain cellular homeostasis. But in inflammation, mitophagy is frequently dysregulated. This results in an accumulation of dysfunctional mitochondria, which results in the propagation of ROS-induced inflammation. However, an area of mitochondrial dynamics that remains to be dissected is the realm of cell-free extracellular mitochondria. In neuroinflammatory models, there is evidence that the presence of dysfunctional extracellular mitochondria may be a signaling agent of neuroinflammation. But how these extracellular mitochondria are released from the cell and their nature in the context of inflammation remains to be elucidated. There is evidence that by inhibiting mitophagy, there is increased release of extracellular mitochondria, which suggests a role for it as an alternative quality control mechanism or a pro-inflammatory signaling mechanism. It's also known that lipopolysaccharides, or LPS, which are endotoxin from gram-negative bacteria, recapitulate the same propagation of inflammation dependent on mitochondrial dysfunction. So here, I use LPS-induced inflammation in Ra264.7 cells as our model system. I hypothesized that LPS-induced inflammation results in an increase in extracellular mitochondria, and I hypothesized that this is due to stalled mitophagy. For my experimental objectives for this thesis, I hoped to evaluate changes in mitochondrial function and oxidative stress due to LPS to confirm that LPS-induced inflammation in raw cells is consistent with the literature. I also hoped to observe changes in extracellular and intracellular mitochondrial content due to LPS and hopefully connect that to changes in mitophagy regulation. I see this thesis as a stepping stone to revealing the nature of extracellular mitochondria and the regulation of their presence in inflammation with respect to mitophagy regulation. To measure changes in mitochondrial function and oxidative stress due to mitochondrial ROS, mitochondrial bioenergetics were evaluated using an Agilent Seahorse XFE analyzer to measure changes in mitochondrial respiration following LPS exposure. Additionally, cells were stained with mitosox red as a marker of mitochondrial ROS to measure changes in oxidative stress due to mitochondrial ROS, and I quantified these changes via flow cytometry. To elucidate changes in intracellular mitochondrial content in response to LPS, I used SDS-Page and Western blot analysis, and I probed for the outer mitochondrial membrane protein VDAC1, the inner membrane protein TIM23, and a matrix protein aconitase as markers of mitochondrial content. To measure changes in extracellular mitochondria, as shown in Figure 2, I collected media from control and LPS-treated cells, and I isolated extracellular mitochondria via differential centrifugation. I stained samples with mitotracker green as a marker of mitochondrial content, and I quantified via flow cytometry. Figures 3 and 4 show that raw cells treated with LPS display a decreased mitochondrial function and increased oxidative stress. Figure 3 shows that LPS caused a decrease in mitochondrial respiration, specifically decreases in maximum oxygen consumption rate and spare respiratory capacity. Figure 4 reveals that incubation with LPS for 24 hours caused a significant increase in mitochondrial ROS as detected by mitosox red median fluorescence intensity. Moving on to changes in intracellular mitochondrial content in response to LPS. Figures 5 and 6 show that LPS exposure results in a decrease in mitochondrial marker proteins. Figure 5 is a representative image of a Western blot detection for mitochondrial proteins aconitase, VDAC1, and TIM23. Beta-actin was used as a loading control since it's a housekeeping protein. And figure 6 reveals that LPS exposure for 24 hours results in a significant decrease in mitochondrial marker proteins as quantified by their respective band intensities, and this was normalized to beta-actin. Moving on to changes in extracellular mitochondrial content, figures 7 and 8 show that LPS exposure results in a significant increase in extracellular mitochondria. Figure 7 is a representative image showing the forward versus side scatter pattern for cell-free mitochondria via FACS. Figure 8 shows that LPS exposure for 24 hours results in a significant increase in extracellular mitochondria normalized to the cell count from which the sample originated from. In summary, in my thesis, I was able to confirm that LPS reduced mitochondrial respiration function and increased oxidative stress due to increases in mitochondrial ROS. Furthermore, I was able to show that following exposure to LPS, there is decreased presence of outer mitochondrial membrane, inner mitochondrial membrane, and mitochondrial matrix proteins, which is very, very strong evidence pointing towards decreased intracellular mitochondrial content. Finally, I was able to show that there is a significant increase in extracellular mitotracker green positive events, which I used as a marker of extracellular mitochondria following LPS exposure. So though I was able to show changes in mitochondrial function, mitochondrial ROS, and mitochondrial content, I was not able to say anything quite yet about mitophagy regulation. Though I observed induction of autophagy following treatment with LPS in separate experiments, this does not necessarily say anything about mitophagy induction, as mitophagy can happen independently of autophagy induction. So my hypothesis still holds. I hypothesize that this decrease in intracellular mitochondria is due to increased extracellular release of mitochondria as an alternative form of quality control due to stalled mitophagy. So future directions include observing Parkin and PINK1 localization to the mitochondria to see how Parkin-mediated mitophagy is regulated in the presence of LPS. I also plan to do mitochondrial lysosome localization studies to confirm or reject my hypothesis that mitophagy is stalled in LPS-induced inflammation. I also plan to study the direct effects of extracellular mitochondria on inflammation. Finally, I wish to move forward with my findings into a neuroinflammatory model. I potentially want to use microglia to study the propagation of neuroinflammation in neurodegenerative disease. This work has so many applications in the realm of acute and degenerative diseases. Thank you for listening to my presentation.